Thank you very much. So Gwenaëlle Lashermes, so you are from the mix laboratory between the university and the Research for Agriculture, Food and Environment organism, and you're going to talk about microorganisms, substrates, interactions during the biodegradation of plant litter in agroecosystem. Thank you very much, Gwenaëlle. The floor is yours.
Yes, thank you. So does it work? Yeah?
Yes. Yeah.
Yes. So yes, so thank you for introducing me and let me the opportunity to present my work. So I'm a research director at INRAE, that is a public research institute in French from agriculture, food and environment. And yes, and I am in a joint unit, the FAR, with the University of France, Champagne-Ardenne. It's FAR, that stands for Fractionation of AgroResources and Environment. So plant litter biodegradation in agroecosystem is a central process for biogeochemical cycle, and in particular carbon and nitrogen cycle. So the residual plant part of biomass that are not exported for food or the transformation of the bioeconomy are restituted to soil and form the soil organic matter that is going to be decomposed or biodegraded by microorganisms. And here we have a lot of interaction between microbes and the substrates. So this process is important. I'm not sure I can change the slide. Yeah. It's important for to recycle the nutrient to the plant, but it also produces greenhouse gas emission, CO2 and N2O, and it's a basis for food web and contribute to carbon. This is an important process to contribute to humus formation and the carbon storage of the organic matter content of soil. It is also important for the quality of water with the leaching of the nutrients. So in the current challenges for agricultural, food and environment, we have to ensure food security and sustainably manage natural resources essential to life, in particular the soil. Reduce global warming, reducing greenhouse gas production, increasing carbon storage, and at the same time producing energy. So these are very, very big challenges, and optimizing the use of carbon and nutrients, in particular for the nitrogen and phosphorus from organic resources in soil is very important to preserve the soil carbon sinks and the better close biogeochemical cycles. That means not to lose nitrogen in the leaching and the greenhouse gas, and also by erosion. So I want to talk about a little bit about numerical models. That are very useful tool for us that we use in our lab for understanding and predicting. So they can help conceptualize understanding the processes, the very complex processes that occur in soil, and also prioritize factors. But some other model can also help to predict how to recycle nitrogen, make a greenhouse gas assessment, pay or remunerate beneficial cropping practices. But the ecology of microbial communities is usually not represented in predictive models. I show you here a very old model here proposed by Hénin and Dupuis in 1945. And this is a model more or less still used today, where you have the plant residues that decompose in CO2 and constitute the thorough organic matter, but the microbes are not represented in this example. So if we want to make confident projection of carbon and the possible feedback of the ecology of microbes on this projection and the way the nitrogen can change this projection, it is important to refine a little bit these tools. So there are different kinds of models. From that, some are used more for understand mechanisms. They can be a little bit complex. Then they can be integrated in a specific context and being a little bit simpler. And then they can be integrated to predict in this context and be incorporated in a decision support tool, or for example, to make some advice for practices or to remunerate the practices or agricultural practices. And this is a progressive and iterative process of modeling in which we revise the integration, acknowledge when the context changes, for example, because of climate, practices, discovery. So in our lab, we are working on all these steps, and my main focus is with the mechanisms here. So let's go a little bit deeper into the mechanisms of plant litter biodegradation. So the organic substrate here is biochemically a complex set of interconnected polymers, and the microorganisms secrete exoenzymes that which catalyze the degradation of this organic substrate into smaller and smaller polymers until the total degradation of the mineral element to CO2 and ammonium here. And the small polymers and the inorganic element can be assimilated by microbes that will multiply and grow. So the biodegradation is the focus of this broader decomposition process, which also include the mortality of microbes and the production of N2 with during nitrification and denitrification of nitrogen. And all these compounds can be incorporated into soil aggregate or interact with soil mineral surfaces, and so being stabilized in soil. And yes, so we are studying all these processes and the constraints that control these processes. In particular, the stoichiometry of microorganisms, which is important for the partition between what they need, what they will assimilate, and what they release. Oops, sorry. So my main focus for today is I want to show you different kind of work on the investment of microorganisms into enzymatic activities to acquire their resources and the impact on the microbial carbon use efficiency. So the carbon use efficiency is the ratio between the carbon that will be used for a microbial growth over the total carbon decomposed. So we first worked on model development, so it was more that theoretical framework with development of a model that is called exoenzymes. Then experimentation, we performed an experimentation with the microorganisms used as an experimental model. And then we test the significance of the indicator and physiological indicators that we define in the field. And we extend, we are ongoing project that in which we extend the concept on a set of terrestrial ecosystem and the nitrogen. So first with the exoenzyme model. So in collaboration with some colleagues from the University of Toledo and New Mexico, we developed this model. This is it simulates the activities of exoenzyme hydrolytic enzymes for the acquisition of carbon here and the acquisition of nitrogen here. This they degrade the substrate that is made of two pools. The one contains carbon and nitrogen, and that could be protein. The second pool is with cellulose, is only carbon. That could be, for example, cellulose. So in this work, we hypothesized that it could be possible to improve the understanding of microbial functioning to represent the energy cost involved in the exoenzyme production, the allocation choices for the microbes in the different enzyme type, and the return on investment enjoyed by the microorganisms. So this model was able to reproduce observation, and that was made by Robert Sinsabo and colleagues in the environmental sample, but I don't show you that today. Then we continue the development with this colleague to develop a theoretical framework for the metabolic cost of lignin degradation. So here the x-axis represents the lignocellulose index, which is the ratio between the lignin over the other compounds of the cell wall in the plant cell. And as the lignin content of cell wall increase, we have a decrease in the degradation coefficient of cellulose and hemicellulose. And we making lignin degradation unattractive from an energy point of view. And above a given threshold, lignin degradation begins to be attractive, not because it brings energy per se, but because it releases sugar. So this framework was the basis to the development of the model, the EASY model, which simulate the partitioning between the hydrolytic enzymes and the oxidative enzymes that degrade lignin. So I skipped that, but after we observe in some field data at which level of lignin does the production of oxidative enzyme begins. Then we performed experiment with a fungus that was and we let it decompose the substrates like leaves, stems, and roots of maize plants, and we measure its assimilation, carbon assimilation, and respiration, and also the enzyme activities. And we conclude that the carbon use efficiency in this case was depending on the ability of the microbes to invest in enzyme and the efficiency of these enzymes. After that, we tried to test this concept into the field. So we have a long-term experiment in France with annual crop rotation with intensive farming, and we compared the conventional tillage and reduced tillage. We added wheat at flowering stage or at maturity stage, and we observed with the two soil tillage practices and usage changes in microbial legacy that ended with microbial community with different carbon use efficiency and enzymes efficiency. So we are extending this. We are extending this concept with a new project here. We have 9 experimental sites, and we compare 2 treatments with 2 different ways of managing nitrogen. And with this symbiotic legacy, you will measure the nitrogen, the carbon use efficiency and nitrogen use efficiency of microbes, and the consequence of the carbon stabilization and the availability of nitrogen. And in this project, we have the modeling work on understanding. Modeling for understanding and for predicting. So I skipped that too. We have another part where we work, in which we work on end-to emission. Yes. And to conclude, I just wanted to say that in my groups, we are very interested in the interaction between different kinds of organic substrates, microbial communities, their exoenzymes, and the different kind of environment. I thank you for your attention.
Thank you very much, Gwenaëlle. Is there any questions? It was quite a good transition with what did Krista just before.
Yeah.
Taina.
I don't know if you...
Taina. Yes, thank you for your really interesting presentation. I would ask, would this modeling, how attractive is lignin usage? Is it possible to calculate in wood, or the systems are in more like systems where lignin is free in the environment?
Sorry, I'm not sure. Could you repeat just the beginning, because it was not clear for me?
Yeah, sorry, I mean that in what kind of systems where lignin is it, does these calculations hold for where lignin is free, or lignin is like inside wood, or like as a complex, in what was the plant litter material where these calculations hold?
I'm not sure I catch everything, but for the are you asking the type of wood, of the type of substrate?
The type of substrate, yes.
Yes, yes. Type of substrate. So with the fungus, yes, yeah. we tried several kinds of substrate, but maybe more crop residue, and also root residue, root litters.
Yeah.
And
okay.
Yes, not we did not work on wood, and I think that this fungus maybe would have preferred wood.
Okay. Yes, that's what I thought. Yeah. Okay.
But for example, with wheat, we put wheat. Wheat is very rich in lignin, so yes, it could be, and he managed to degrade it.
Oh, okay. Thank you.